Welcome to G Fun Facts Online, your go-to source for the most intriguing discoveries from the world of science and beyond. Today, we're diving deep into a tiny terror that causes worldwide chaos year after year, the influenza virus. You know the drill. The seasonal flu, the annual shots, and sometimes those shots just don't seem to hit the mark. But what if we could change that? What if we could create a universal shield against this master of disguise? Stick around, because we're exploring the cutting-edge science aiming to do just that. Influenza, or the flu, isn't just a nasty cold. It's a formidable global health challenge. Why? Because this virus is a shapeshifter. It mutates rapidly, constantly changing its appearance to evade our immune system. This is why traditional flu vaccines often struggle. They're designed to target specific strains, but if the virus changes too much, the vaccine can become a mismatch, offering reduced protection. It's like trying to hit a moving target that's also changing its shape. This frustration has fueled a massive research effort into something called monoclonal antibodies, or mAbs, with the ambitious goal of creating universal therapies and vaccines that can protect us against a broad spectrum of flu viruses. So what are these monoclonal antibodies, and how can they help? Think of them as highly specialized guided missiles for your immune system, engineered to neutralize a wide array of influenza A and B viruses. Scientists are employing some truly ingenious strategies. One major focus is targeting parts of the virus that don't change much. These are called conserved regions. The influenza virus has a protein on its surface called hemagglutinin, or HA. Imagine the HA protein like a mushroom. It has a head region, which is highly variable and the main target for current vaccines. But it also has a stem region, which is much more stable and similar across different flu strains. Scientists have developed broadly neutralizing antibodies, or BNAbs, that specifically target this HA stem. These antibodies are like supergrips that clamp onto the stem, preventing the virus from fusing with our cells, which is a critical step for infection. For instance, research highlighted in January 2025 showcased exciting BNAbs like Fi6, which can impressively bind to all 16 known influenza AHA subtypes. Other heavy hitters in clinical development include Medi 8852 and Vis 410, showing potent effects. Another one, MHAA 4549A, has proven well-tolerated and effective in reducing how much virus-infected people shed, a big step in controlling spread. But scientists aren't putting all their eggs in one basket. Even the highly variable HA head region has some hidden conserved spots, like the receptor binding site, the very part the virus uses to latch onto our cells. Antibodies like CH65 and fragments derived from S139-1 can target these sites, neutralizing multiple H1N1 strains. Researchers have even found new targetable spots hidden deep within the HA structure. And it's not just about the HA protein. There is another glycoprotein on the virus surface called neuraminidase, or NA. It's less abundant than HA, but still a good target. Recent discoveries of protective antibodies that recognize conserved bits on NA from diverse flu A and B viruses have brought NA back into the spotlight as a vaccine target. Some of these NA-targeting mAbs have even shown they can work therapeutically in animal models if given up to 72 hours after infection. That's a huge window. Beyond just where to target, scientists are also getting creative with how they engineer these antibodies. They're building multi-domain antibodies that can grab onto multiple viral targets at once, making it harder for the virus to escape. Then there are nanobodies, which are tiny antibody fragments derived from animals like camels and llamas. Their small size and stability allow them to access hidden nooks and crannies on the virus that normal antibodies can't reach. Gene transfer techniques encoding these engineered antibodies have even shown cross-protection against both influenza A and B viruses in animal studies. A major breakthrough. Scientists are also experimenting with different antibody formats. For example, IgM antibodies are naturally large molecules with multiple binding sites. Engineered IgMs can effectively blanket the viral surface, preventing it from engaging with our cells. This multivalency also helps overcome viral escape mutations. Similarly, polymeric IgAs, especially those found in our nasal passages and boosted by intranasal vaccines, have shown enhanced broad protection, right where the virus often enters our body. Even the tail end of the antibody, called the Fc domain, can be tweaked. Modifying this part can boost the antibody's ability to call in other immune cells for backup, leading to more effective viral clearance. And underpinning all of this is the power of modern computing. Advanced computational tools and a deep understanding of how antibodies and viruses interact at a molecular level are allowing scientists to rationally design antibodies with pinpoint accuracy, improving their strength and breadth. And let's not forget the mRNA technology that became a household name with COVID-19 vaccines. This tech is now being harnessed for influenza. mRNA vaccines can be rapidly adapted to viral mutations and can be designed to make our bodies produce the very antigens that elicit these powerful, broadly neutralizing antibodies. Multivalent mRNA vaccines are in development, encoding multiple HA antigens to offer broad protection. A great example is Moderna's mRNA 1010, which targets 4 seasonal influenza strains in one shot. Now, despite all this incredible progress, there are still hurdles to overcome. Producing these sophisticated mAbs can be expensive, making them less accessible in low and middle-income countries. Ensuring that the protection they offer is long-lasting and figuring out how to stay ahead of the virus' ability to mutate and escape are ongoing research priorities. There's also a theoretical concern called antibody-dependent enhancement, or ADE, where antibodies might, in rare cases, make the disease worse. While this hasn't been a major issue for influenza B and ABs, it's something scientists carefully monitor, but the future is undeniably bright. The development of these broadly neutralizing antibodies is a cornerstone in the global strategy to achieve universal influenza protection. Whether delivered directly as a therapy or elicited through next-generation vaccines, these engineered molecules hold the promise of transforming how we prevent and treat the flu. We're talking about longer-lasting immunity, better preparedness for seasonal epidemics, and a stronger defense against future pandemics. The relentless innovation in antibody engineering, combined with new vaccine platforms, is paving the way toward a future where the threat of influenza is significantly diminished. That's all the time we have for today on G Fun Facts Online. We hope you found this glimpse into the future of flu protection as fascinating as we did. Join us next time for more amazing facts and discoveries. Thanks for listening.